Today we're going to be discussing action potentials and the movement of both water and ions across membranes, and how neurons or other cells throughout the body can use the movement of ions across the membrane to establish a membrane potential and then change that membrane potential and cause waves of depolarization or positive charge that move down neurons or cells in general to create what are functionally called action potentials, and that can enact a change from one side of the cell to another side of the cell and influence signaling systems. So up here towards the top of this diagram, I have a cell, and this is a eukaryotic mammalian cell, so it lacks a cell wall. And the issue with this cell is that it's suspended within a watery environment, so there's a lot of water outside the cell. And because most of these cells, especially mammals, are in warm environments, there's a lot of kinetic energy. So these molecules are always moving around and they bounce off each other. And if I have a high concentration of water outside the cell relative to the inside of the cell, water will tend to move from high concentration to low concentration, moving from the outside to the inside, because a lot of these molecules on the inside of the cell, like these proteins and nucleic acids and different monomers like amino acids or ions, like these guys could be represented as ions, they're occupying space within the cell, and that space where water is not there. So I have more water out here relative to the inside. So water will move from by a concentration gradient from high to low concentration into the cell typically. And the issue is, if there's a net gradient of water moving into the cell, that'll increase the overall concentration of water or the amount of the volume of water within the cell, causing a hydrostatic pressure to push on the outer membrane, push on this membrane, causing the cell to swell up and potentially break apart. And so these cells solve this problem by trying to even out the amount of solutes on the outside and the inside. And what that means is they've developed mechanisms for actually pumping out ions like sodium. So I typically have some sodium within the cell. Sodium is a solute in this case. It's contributing to the osmolarity, which is just a definition of the amount of solutes per volume of fluid. And so these sodium ions are pumped out of the cell actively, which means that I'm using ATP, an energy source. I'm using ATP, converting that ATP into ADP. In the process, I'm moving sodium from the inside of the cell to the outside of the cell, and sodium tends to have a higher concentration outside of the cell because I'm actively pumping it out. So I'm pumping sodium from a low concentration within the cell towards a high concentration. So it's going against its normal concentration gradient, which means that it takes energy. It takes energy in the form of ATP, hydrolyzing that phosphate and producing ADP. And so I'm pumping out a bunch of sodium out here. Sodium has a positive charge. And then at the same time, I'm pumping potassium, K+, into the cell. But what's unique about this is I'm pumping 3 sodium out and 2 potassium in. So overall, I'm pumping more positive things out of the cell. So overall, the outside of the cell is starting to become more positive relative to the inside, which is becoming more negative. And this is important because it establishes what's called membrane potential, where the inside of the cell, right along the barrier of the membrane, becomes relatively negative. And due to the activity of these, what are called sodium-potassium pumps, using ATP to pump 3 sodium out and 2 potassium in, it establishes that negative membrane potential, and it establishes it at around -70 to -90 millivolts. And so normal resting cells will have a membrane potential around -70 to -90 millivolts, but that can change depending on what this cell lets in. So for example, if I were to like draw like a little channel right here in the membrane. And let's say I let in an ion like calcium, which has a positive 2 charge, 2 plus charges, and I let calcium into this cell. What that does is that'll start to make the membrane potential more positive. It'll still be kind of in the negative range depending on the amount of calcium that's moving in, but it starts to become a little bit more positive in this little local area, which can influence proteins because proteins have a 3D shape that can be changed by changes in voltage. So if I have a protein like this, and there's different regions of this protein that are positive and negative and positive and negative, and now I introduce something like calcium that's around, calcium with that +2 charge can actually change the shape of that protein. It can bind to different regions of that protein. And if I change the shape of the protein, I can change the function of the protein. And so this diagram is important for understanding the action potential. And I'll erase some of this stuff in here. It was just meant to show that solutes can play a role in the movement of water because water is moving with a concentration gradient, as are other molecules. And so within the cell, because of that sodium-potassium pump, I start accumulating a lot of potassium inside of the cell because I'm actively pumping sodium out of the cell. But at the same time I'm pumping sodium out of the cell, potassium is moving into the cell. And so I have a higher concentration of potassium inside of the cell. And potassium will reach something called a compromised equilibrium, where I have a lot of potassium inside the cell. All of these little ions are potassium. And then I can have something called a leak channel, which is just like a little pore in the membrane. So it's similar to this guy right here, but a little pore in the membrane that allows potassium to kind of just move with its normal gradient. And so if I have a lot of potassium inside the cell, potassium will move out following a concentration gradient. But the unique part of this is, if I have potassium on the outside of the cell, because it just moved out, potassium has a positive charge. And remember, we established a relatively negative membrane potential around -70 to -90 millivolts. And so potassium's moving out with a concentration gradient. It's also moving in with a charge gradient because it has a positive charge. So potassium will move in because it's a positive moving towards a negative. Positives and negatives are relatively attracted to each other. So potassium's moving in following a charge gradient or an electric gradient, electrochemical gradient, and the potassium's moving out following a concentration gradient. But overall, the strength of the movement, moving out, concentration in with charge, is about equal. And so there's not really a net movement of potassium. So potassium is still moving back and forth because it has kinetic energy. It's attracted via from the outside moving in by a charge gradient. So overall, it's still moving, but there's no net movement. But let's say I were to change the membrane potential and make this membrane potential a little bit more positive. And then you may ask yourself, what is going to change in terms of the movement of potassium? Overall, the movement of potassium moving from the inside out probably won't change much because I'm not really changing the concentration of potassium on the inside by changing the charge. So potassium's moving out with full force, but the drive for something with a positive charge to move towards something that's now more positive, because I changed the membrane potential, is less. So I can kind of erase part of this arrow, and you can see that the movement out is pretty strong, but the movement in isn't quite as strong. So the net movement of potassium would be out of the cell when the membrane potential becomes more positive. And that could be as simple as a change from -70 moving towards something like -50 millivolts. It's changing the membrane potential slightly. It's still negative, but it's more positive than -70 or -90. And so if I scroll down, this is a little diagram that's supposed to depict kind of the overall morphology of a neuron. And so this region of our neuron is like the dendritic region, where you'd have like the little nucleus right here. And then starting right here, this would be called the axon hillock. Hillock. And then from that axon hillock point all the way down towards the end, this box right here would be considered the axon of the neuron. And then right where the axon ends, all the way to the very end of the neuron, this is like the axon terminal, or the terminal region of the neuron. So this side of the neuron typically receives signals from the environment. It could be other neurons. It could be dissolved signal molecules in the blood. And then this part of the neuron transmits the signal. And then this part of the neuron is responsible for releasing something to stimulate an effector, which could be another neuron. It could be a muscle. It could be a gland. But overall, it's about how do I relay a signal from one side of the neuron to another side of the neuron. And so this dendritic side of the neuron, this part right here, will have channels that are called ligand-gated ion channels. And it's important to define what a ligand is. So a ligand is typically a water-soluble signal molecule. So some molecule that's dissolved in water. It could also be a non-water-soluble signal molecule that's able to dissolve in lipids or fats. But usually in terms of neurotransmitters, it's water-soluble. And so let's say this is our ligand right here. It's just a little signal molecule. And signal molecules can come over and they can bind to receptors that have a complementary shape that can fit the shape of the ligand. So this signal molecule can come over and bind to a receptor, and it can change the shape of the receptor, and the receptor is physically attached to a channel. And a channel allows something to move from the outside of the cell into the cell. So it's not necessarily letting the ligand inside of the cell, but what's happening is the ligand's binding here to this receptor, changing the shape of the receptor, which changes the shape of the channel, which can either open it or close it, or allow it to be more permeable or less permeable to something moving from the outside into the cell, or something moving from the inside out of the cell. And so in this example, this would be considered a ligand-gated sodium channel. So this is a ligand-gated sodium channel, Na plus channel. And the ligand in this case is typically something called glutamate, which is a stimulatory neurotransmitter. So I'll put in parentheses glutamate. And it's just a molecule that's released by neurons that tends to bind with ligand-gated sodium channels. And if glutamate binds right here, it allows this ligand-gated sodium channel to be permeable to sodium. So it allows sodium to move from the outside of the cell into the cell, which will start, because the sodium has positive charge, it'll start to depolarize the membrane or make it more positive. So it's making the membrane more positive when sodium moves in, because remember, we're actively pumping sodium out of the cell. So I have a really high concentration of sodium on the outside of the cell. So lots of sodium out here because I'm actively pumping it out. And so if I open a gate, sodium will move in following both a concentration gradient, because I have a lot of sodium out here. All these little molecules are sodium, but also following a charge gradient, because sodium has a positive charge and it's moving towards the inner membrane, which typically has a more negative membrane potential or a little negative charge relative to the outside of the cell. So sodium's rushing in the presence of glutamate, because glutamate is binding to the glutamatergic receptor on a ligand-gated sodium channel. So sodium's entering, coming in, it's starting to depolarize the membrane or make it more positive. You can see these little negative charges are becoming now more positive in this little local area. And the interesting part is, if it gets positive enough, if enough sodium enters the cell where it reaches this region called the axon hillock, this is a region where I transition from having a bunch of ligand-gated sodium channels and ligand-gated chloride channels, or just ligand-gated channels in general in this region, towards a region where I have voltage-gated channels. So voltage-gated channels are different from ligand-gated channels because they're sensitive to voltage. They're not sensitive to signal molecules typically. And so this right here, this channel, is an example of a voltage-gated sodium channel. And a voltage-gated sodium channel is sensitive to voltage, when the voltage becomes more positive. And it's typically in this conformation where I have a top gate called the activation gate and a bottom gate called the inactivation gate or the deactivation gate. And the top gate's usually closed. So nothing's really moving out, nothing's really moving in because this top gate's closed. But when the membrane becomes a little bit more positive, it's normally negative, but when it becomes a little bit more positive, it'll cause this top gate to flip open. So I'll erase this top gate. It's now in an open conformation. It's wide open, which allows sodium, Na+, to move from the outside of the cell into the cell. So sodium's rushing in, following both a charge gradient because it's positive, moving towards a more negative region, and following a concentration gradient because I have a lot of sodium out here. Lots of sodium ions on the outside of the cell. So they're moving in, following concentration gradient, moving from high to low. And so that will further depolarize the membrane. It'll make the membrane even more positive, which causes the exact same thing to happen to this next voltage-gated sodium channel. So the top gate, the activation gate, will flip open. This gate flips open, which will allow sodium to move from the outside of the cell into the cell, which will start depolarizing the membrane or making it more positive, which will cause this next channel to open, this next channel to open, all the way down the length of the cell until automatically the bottom gate will just close. So the top gate flips open, and briefly after, the bottom gate will automatically close. So there's a brief window in which sodium will rush in, but as soon as it does, the bottom gate closes quickly, and so sodium can no longer rush in. There's a blockade where sodium will reach this bottom gate or this deactivation gate and be inhibited from moving further. And the same thing can happen to this channel where now this is closed. The same thing can happen over here where the top gate, the top gate flips open, and then the bottom gate will quickly shut. So quickly shuts. But overall, you're getting a wave of positive charge that's moving down this axon, down the length of the neuron, because sodium is rushing in, and it's causing positive charges to accumulate along this membrane in the little local area, but that causes the opening of the next voltage-gated sodium channel, which causes sodium to move in, depolarizing this little local area, which causes the next one to open, depolarizing it. But you may ask, if this occurs, it'll cause a wave of depolarization, but that can only happen once because that'll just cause these channels to be in this conformation. How do I reset the conformation in order to cause this to happen multiple times? So that's where something called a voltage-gated potassium channel comes in. So these guys, purple guys, are called voltage-gated potassium or K+ channels. And they're also sensitive to changes in voltage. And what that means is when, so typically the membrane potential is pretty negative, but when sodium rushes in, it starts to make the membrane potential a little bit more positive. And that membrane potential that's getting more positive will cause the opening of a voltage-gated potassium channel. And if a potassium channel opens, it allows potassium to move from the inside of the cell to the outside of the cell. So potassium tends to be in higher concentration on the inside of the cell because of the function of these sodium-potassium pumps, which are moving sodium out but potassium in. So overall, usually I have more potassium on the inside of the cell, and potassium will move following a concentration gradient out of the cell. But remember, potassium also has a positive charge. So once potassium's outside of the cell, it has a positive charge, and it'll tend to move back into the cell following a charge gradient. But now I've changed the membrane potential from being more negative, which would attract potassium inside the cell, to being now more positive. And if the membrane potential is more positive, there's less of a drive for potassium to move from positive to something that's now positive. So the overall net movement of potassium would be out of the cell. And as potassium moves out of the cell, as each one of these little potassium ions is starting to move out of the cell like this, they're also taking a positive charge with them. Potassium has a positive charge, so I'm removing an ion, K plus, out of the cell. Therefore, I'm losing something that has a positive charge, which will cause the membrane to become more negative again. And this is functionally resetting my neuron back to resting membrane potential. So now it's becoming more negative on the inside, more negative. And all of these channels, both the voltage-gated sodium channels up here and the voltage-gated potassium channels down here, are all sensitive to voltage, where they open with depolarization, but they close with repolarization or a negative charge. And if I'm repolarizing this neuron right here and making it more negative, these voltage-gated potassium channels, these purple guys, will start closing. So they're that little channel opened, now it closes because it's more negative again. And so these voltage-gated potassium channels, these guys on the bottom, are responsible for resetting the neuron. And they also help these voltage-gated sodium channels reset themselves too. And so what happens is when potassium leaves the cell, it causes this top gate to close. Causes the top gate to close. And then it causes the bottom gate to open again. So this bottom gate will open again. So now these channels, voltage-gated sodium channels, are reset to their original position that they were before I encountered a ligand, before that ligand bound to a ligand-gated sodium channel, before I let sodium into the cell, which depolarizes this little local area, and before I cause the opening of voltage-gated sodium channels. And then you may ask, like, how does this wave of charge that I'm causing due to the movement of sodium into the cell actually enact an effect on something over here? Because the whole idea is that I have a signal occurring over here or something that's being detected by this neuron, and I'm relaying that signal through a wave of positive charge to the end of the neuron so that the neuron can have an effect, or the cell in general can have an effect on something over here. So I encounter a signal and I cause an effect in order to respond to whatever the environmental or the internal signal would be. And so at the very end of your neuron, you have what are called voltage-gated calcium channels. So voltage-gated calcium, Ca++ channels in brown. And so these guys are voltage-gated calcium channels. They're also sensitive to changes in voltage. And what happens is when you have that wave of depolarization caused by the movement of sodium inside of the cell from the outside, it causes a depolarization or that positive charge, which will also affect these voltage-gated calcium channels, causing their little channel to open. So it opens the gate on these voltage-gated calcium channels. It flips that little gate open, as well as the gate over here. And it allows calcium, which is typically found outside of the cell. Calcium is another ion that's pumped out of the cell using ATP. Calcium can then move into the cell, but calcium is unique because it can bind with proteins and change their shape, therefore changing their function. And so what happens is you have these little vesicles, so these little cells within a cell. So this is considered like a membrane right here. All this is considered membrane. And these little vesicles hold molecules that are typically called neurotransmitters if they're released from neurons, but they could just be signal molecules if they're released from other types of cells. And so within this little vesicle, there's a bunch of signal molecules that are present inside of it. And calcium will rush in and it'll bind to proteins that sit on this little vesicle. And they're typically called snare proteins. And if calcium binds to these proteins, imagine this is like a little calcium molecule that's bound to the protein. It'll change the shape of the protein and cause the protein to move this little vesicle towards the outer membrane and fuse the membrane of the vesicle with the outer membrane, which allows this neuron to release those signal molecules, those little red signal molecules, out into the space that's outside of a cell or into interstitial space, or sometimes it's termed synaptic space. So overall, this was kind of an introduction or just a diagrammatic way of drawing out action potentials and understanding like the movement of ions, potassium, sodium being pumped out of the cell actively, and how different channels can influence the wave of depolarization of the cell. But there's also a different type of ligand-gated channel called the ligand-gated chloride channel, which introduces a no vote to the system. So typically ligands like glutamate that bind to a ligand-gated sodium channel and allow sodium to move in, they're kind of a yes vote for whether or not a neuron will fire an action potential, which is because sodium has a positive charge, so it'll move into the cell. It'll start depolarizing the membrane, so it makes it more positive. Whereas a no vote would typically be something like chloride, where you have another ligand, so a signal molecule. Typically in the central nervous system, this signal molecule is called GABA, or in the brain, it's called GABA. In the spinal cord or the caudal part of the nervous system, it's called glycine, Glycine. And this ligand will come over and bind to a receptor on a ligand-gated chloride channel. And instead of causing this channel right here, this guy right here, to be permeable to sodium, it's now permeable to chloride. So chloride ions have a negative charge. So chloride will kind of counteract the effects of sodium, where if chloride enters the cell following a concentration gradient when this channel is open, chloride will move in and it'll make the membrane more negative. So if it makes it more negative, it'll prevent these voltage-gated sodium channels from opening. Therefore, it'll prevent the wave of depolarization or positive charge. Therefore, it's a no vote telling this neuron not to fire, whereas these ligand-gated sodium channels are a yes vote because they're causing depolarization or the propagation of a positive charge in this little local dendritic area, which will cause the opening of voltage-gated sodium channels at the axon hillock, which will spread the wave of positive charge all the way to the end of the neuron, potentially releasing a vesicle that contains neurotransmitters into the synapse, causing an effect on the next neuron. So there's both yes votes and no votes, which cause some integration to occur. And overall, this is just a depiction of the movement of ions and the basics of an action potential.